Hello, welcome to another video from the conscientious biologist, Ben Gallagher. This one's the 7th one in the genetics and inheritance playlist, and it follows on very, very closely from the last one on genetic inheritance. Now, this one serves two main purposes. The first one, and probably most important, is to really reinforce the theory of genetic inheritance and the mechanisms by which it happens that were covered in the last video. This one looks at a few different ways that can be shown. In particular, we look at family tree diagrams. The second purpose of this one is it covers a couple of specific genetic conditions that are part of the GCSE syllabus and that you need to know about. As I've said, it is from the GCSE syllabus, and please do subscribe to my YouTube channel if you haven't done so already to keep updated on new releases. Thank you. Okay, so first off, let's look at a really classic example that's wheeled out every year in schools, and to look at eye colour. Now, eye colour is a classic example, but it's factually misleading quite often the way that it's put across to you. And sometimes in some textbooks, certainly some of the older textbooks, it's a little bit misleading because you may have already been told, or you may have seen in books, that there's brown eyes and blue eyes, and we look at the genetics of that. When of course, there's loads of different shades of eye colour. Look at all the ones in the chart up above my head there. Right, even from sort of opposites, you can have totally white irises, or you can have totally black irises where the pupil and the iris, that's the coloured bit, there's like no difference between them, so it looks like people have just got a giant pupil. You can have yellow eyes, you can have pink eyes in albinos, for example. So there's loads of variation. So there's clearly something more complex going on here than anything really simple, and that's because there are actually 8 identified genes that control the proteins that get put into the irises in your eyes. The irises are the coloured bit, it's the proteins in there that reflect light to give you the colour that you see. Now, to make things simple though, we're going to focus on one of those 8 genes, the OCA2 gene, because that one controls the brown and blue colours. So this is closest to the simplified and classic examples. We're just going to stick to that. So if we're looking at the 2 alleles of the OCA gene, you've basically got a brown allele and you've got a blue allele. We'll use a big B and a little b to symbolise them, the capital letter showing the dominant allele and the lowercase letter showing the recessive allele. Now, if we put that into genotypes, we can see that of the 3 genotypes, homozygous dominant, heterozygous and homozygous recessive, we get these 3 genotypes. Okay, remembering heterozygote has both, and the homozygous ones are just one, and whether it's the dominant or recessive. When we look at the phenotype that's expressed from the genotypes, what actually ends up happening, because of the dominant allele, in a homozygous dominant, you get the dominant phenotype, the brown eyes, but you also get it in the heterozygote, because your cells will always read the easier allele. And the dominant allele is more easily turned into that messenger molecule that goes to the ribosomes, from which it can make a polypeptide sequence. It's only the homozygous recessive that will give you the blue-eyed phenotype, because it's only got blue alleles. It has to read one of them, so you're going to get the blue eyes produced. So if we look at something more similar to what you'd get in the exam, it might ask you to show how 2 brown-eyed people can have a blue-eyed child. As always, please, in your genetics questions, set it out in this same way. Think about what the 2 parents were, and then those 6 levels. The parent phenotype, genotype, the gametes that they produce by meiosis when they separate their 2 copies back into individual sets. Then once you cross them, what will be the offspring genotype and phenotype and the ratios. Now let's fill in all the information we can from the limited bit of information they gave us in the question. How 2 brown-eyed people can have a blue-eyed child. So we can put in the parent phenotype. We know they're both brown, it's told us that. We know they're going to have a blue-eyed child, so we can put that in the phenotype. Now, we can discern from that some key things using this information above me. We know that that blue-eyed child has to have the homozygous recessive phenotype, because that's the only one that gives it blue eyes. So we can fill that in. We know it's the little b genotype to give that phenotype. Now, from that, we can work out the parents, because if the child has 2 little b's, well, it got those from each of its 2 parents. So both parents had to have the little b, the blue-eyed allele. They both had to have a copy to give to their child. But both parents have brown eyes, so they both have to have a big B to give them their brown eyes. So from that, we know that both parents have to be heterozygous. Now, as soon as you know that, you can work out what the gametes can be. You can put them into your Punnett square. You can see that each of them have a big B and a little b in their gametes. So when you work out the cross, you get these: one homozygous dominant, two heterozygotes, and of course the homozygous recessive blue-eyed child that we already knew about. So when you convert that into the phenotypes, you get those, which gives you your 3:1 ratio. Okay, so that blue-eyed child there, produced by those parents, by that mechanism, that proves the question. It shows how the two brown-eyed people can have a blue-eyed child. If you want to explain it in words as well, you could just say this. So if both parents are heterozygous, that's the key thing. You've identified the parents must be heterozygotes. Then they both carry the blue allele. That's often worth a mark in the exam. If the offspring gets a blue allele from each parent, it will have blue eyes. And then you could say, because of the ratio, that there'd be a 25% chance of this happening. With the grid, with the information in the blue box over there, and this written paragraph, you're almost guaranteed full marks if the question was just show how two brown-eyed people can have a blue-eyed child. Okay, so let's look at our next example. This is cystic fibrosis, and this is one that's in your specification, so there's details you need to know about this. They might just say in the exam, what is cystic fibrosis? So it's a recessive genetic condition, and it affects cell membranes, preventing the movement of certain substances. Now remember, a cell membrane holds the cell together, it keeps the inside, but it also controls some of the things that can come in and out. So if you've got a faulty protein produced by a faulty gene in the membrane, some things aren't going to be able to be moved in and out of those cells. Now, that impacts many organs, because all of your body is made of cells, but particularly there's an impact of cystic fibrosis on the reproductive, digestive and respiratory systems. Those are systems that have very key things that they need to move in and out of their membranes. Now that gives symptoms in those systems. The first one in the lungs, the most noticeable one, is a sticky, choking mucus that builds up in the lungs. It has to be coughed out every day. They have sort of pummeling massages on their chest and back to try and free up that mucus to cough it up. So that's quite a debilitating thing. Secondly, mucus can clog up the pancreas. Now remember from the digestive system lessons, the pancreas has a major role in secreting digestive enzymes into the duodenum. If it's clogged up with mucus, because it can't move that mucus properly, those enzymes aren't going to be released into the digestive system, so food isn't going to be digested properly, so they're not going to get the right level of nutrition. So you might find someone with cystic fibrosis is quite malnourished, doesn't get a lot of nutrition from their food, simply because they can't digest it properly. The third symptom is a buildup of materials blocks parts of the reproductive systems, which can lead to infertility. So quite often people with cystic fibrosis can't have children of their own. Those are key facts you need to know about cystic fibrosis, but let's look at it genetically. Okay, so the gene is for a membrane transport protein that can move through the membrane. There's two alleles. I'm going to call these regular and faulty. There's all manner of names you could say to it: normal, abnormal, properly formed, malformed. It doesn't matter how we're talking about the protein, but I'm talking about a regular protein or the faulty version of the protein. We'll give that a capital R and a lowercase r to denote the dominant and recessive alleles. Now, we're going to look at this one using one of these. This is a family tree diagram or a heritage diagram. And what we've got are 3 generations of a family there. At the top, we've got the first generation. So that could be like grandparents, if you like. Below that, the second generation. And below that, the third generation. So like grandparents, parents, and your generation there. Now what you've got, if you look at the blue lines that link them, lines going down for individual link them as a couple who then go down and have a child. Okay, so in the first generation, you can see over there, blue lines coming down from them. So that might be the grandmother and grandfather when they had their child that comes down to that individual. That one over in the middle there then had children with the next one to the one next to them, which comes down to our final individual in the third generation. That's the circle on the diagram. So hopefully the layout of these diagrams makes sense. So let's get rid of those labels and bring up the key, because these diagrams always have a key to show you something about the individuals within them. On mine over here, I've got the squares representing healthy people, and I've got the circles representing people who suffer from cystic fibrosis. Now think about what we've said in the genetics already. It's a recessive allele. So to have cystic fibrosis, you must be homozygous recessive, because if you had even one copy of the healthy allele, because it's dominant, you would be healthy. So what we're saying here is that circle at the bottom must be homozygous recessive. The rest of them could be heterozygous or homozygous dominant. As long as they've got at least one healthy allele, they're going to be healthy. But let's look at some exam questions based on this. So if the individual marked with an A, so I've put a red A over there in generation 1, if they're homozygous dominant, deduce all the other genotypes in the family tree. Now that doesn't sound like they're giving us much information to try and work out 5 people, but it's all that we need because of the other information we can deduce. As a second part of this question, they're going to ask what was the probability of individual B, which is that 3rd generation one, being born with cystic fibrosis. So what was the probability? So let's apply what we know. They've told us that individual A is homozygous dominant. So we can fill that in. They're big R, we know that. If they've only got big Rs, they've only got big Rs to give to their offspring. That's straightforward. We can also put in individual B because they are a cystic fibrosis sufferer. They're in a circle, so we know that. And the only way they can have it is if they're little r, they're homozygous recessive. So we've already got 2 built in there. Now, the only way individual B can have it, given the parents don't have it, is as if the parents both had a big R, making them healthy, but also had a little r, carrying the little r, because they will give that to the final offspring. Okay, so it's just like the blue-eyed child and the brown-eyed parents on the last example. We've got 2 heterozygotes contributing to give a child who's going to have cystic fibrosis. Now we can also, though, fill in the last individual up there because we know they don't have cystic fibrosis, so they've got to have at least one big R. And you might be going, well, they could be homozygous dominant or heterozygous. But actually, the child of the 2 people in generation 1, we know is heterozygous because of the child they had. They must have got a little r allele from somewhere. They definitely didn't get it from individual A because we know they're homozygous dominant. So their little r must have come from the other parent. So actually, just applying a bit of logic, we've answered the first part of the question and we've allocated every single individual on there a genotype. So let's look at the second part of the question and look at what probability B had. And we can do this with the genetic cross. Same really as the last one. We know it's 2 heterozygous parents. So when you do the cross, you get these offspring. When you work out the phenotypes of those, you've got 3 that are healthy and just that last one that was cystic fibrosis. So it was actually very unlucky here that the 2 individuals in generation 2, the 2 heterozygotes, had that child, because you get a 3-to-1 ratio with 2 heterozygotes, which means there was only a 25% chance of that child having cystic fibrosis. So that answers the 2 questions and hopefully explains to you what a family tree diagram is, and I've given you the information you need about what cystic fibrosis is. Okay, next example that's specifically on your course and you need to know this is this one. It's called Polydactyly. This is a dominant genetic condition. Have a look at the picture above me and see what's wrong. Got an extra finger. Okay, Polydactyly, there's many forms of it, but the result is extra digits. Those could be fingers or toes. The picture there up above me, that's quite an extreme example, loads of extra toes. But Polydactyly is just extra digits that's formed. It's just a little error in the development that means instead of sort of bits of your hand like that going, yeah, make a finger there, and there, and just those 4, it goes, well, make another one there. Or sometimes, a relatively common one, you might get a thumb growing out that way as well as out that way, and you've got sort of a double thumb. There's lots of ways that polydactyly can manifest in an individual. But the basic genetics is that it's the creation of an extra digit. It's almost always the same on both sides of the body as well, symmetrical. So if you've got it on one hand, it will be on the other hand of both feet, and so on. Okay. Now the curious thing about polydactyly is it's a dominant genetic condition, as opposed to cystic fibrosis was a recessive one. Now what that means is you only need 1 copy of the polydactyl gene to result in polydactylism. So the alleles can be affected by polydactyl, or not affected or normal. And I don't mean normal, I'm not trying to say normal, that nobody's normal, we're all mutants and mutations from each other. That's how evolution works. I'm just saying the normal number of fingers and toes as opposed to the polydactyl. So I'm giving a capital A for the affected polydactyl individuals and a lowercase a for the normal allele, the non-polydactyl allele. Now, if you think about that, it does mean you only need one dominant allele to have the condition. So in the exam, they might say, explain why two normal or non-polydactyl parents cannot have a polydactyl child. Now, hopefully that should be straightforward to you, given what I've already just said, that if they're normal, then their genotype must be homozygous recessive. It's the only way you can be normal or non-polydactyl is to have the two little a's. Well, if the parents have only got little a's to contribute, then their gametes are only going to be little a's. The offspring will all be homozygous recessive as well. There's no other option. The offspring will be normal, will be non-polydactyl. So that should be quite straightforward. So let's look at another question and link it to another family tree diagram to complete the genotypes in the family tree, assuming only one of individual A's, that one up there, only one of his parents was polydactyl. So if only one of A's parents was polydactyl, the other one wasn't. So we know the other one, not shown on this chart, okay, a layer up above this, one of them must have been little a, little a. The other parent could have been big A, little a, or big A, big A. But we certainly know only one of their parents had polydactyly. Okay, so let's fill in what we can here. Well, straight away, given that with the key that I've just put above me, the square is the normal phenotype, non-polydactyl, the circle is polydactyl, we can straight away fill in all of the squares. So most of these we can do instantly, because if they don't have polydactylism, they have to be homozygous recessive. So all we're then looking at is some of the probabilities and how it changes. Now, if you look there at what we've got, we can make some assumptions. If we know that individual A has at least one capital A because they've got polydactyly, and we know that their child has polydactyly, then we can quite realistically suggest that both of them are heterozygotes, because individual A must give an A onto his, a capital A onto his child, but the one in the second generation there must have got a small a from the other parent, so they're definitely heterozygote. Now, because of that, we can look at the offspring down here, and we can say in this final generation closest to me that that one in the middle must also be a heterozygote, because look at the two parents in generation 2. One of them was homozygous recessive, so they have to have given a little a to their offspring, the circle in the middle. The other one must have given them their capital A, so you know they must be heterozygous as well. The only one of these that we couldn't deduce directly was A itself, but if A itself, if it would told us that A had only one normal parent and one polydactyl, then from the normal parent they must have got the little a, making them heterozygous as well. It's really hard for me to just describe that. Normally when I teach that, it's loads of questions. I can listen to people, I can reform it, I can go over it. I don't have that luxury in videos, but re-watch it if any of that doesn't make sense. But it's like a detective story. You're just trying to work things out based on the available information. Now, if I add an extra little section on here, if I say that this one over here in the third generation that we know is a normal non-polydactyl individual, if they have children with another normal non-polydactyl individual, can we identify the mystery genotype? Hopefully, if you've really been concentrating and you followed this, you should be going, yeah, that's absolutely obvious, because it's exactly the same as the cross that we had over there. It's just me reinforcing it. If you've got two non-polydactyl individuals, they've only got recessive alleles to pass on, so they're going to have a recessive individual as their offspring. Okay, last example, we can go quite quickly on this one. This one's quite straightforward. We're talking about gender here. Now, gender has obviously become a massive issue and widely debated over recent years. So I'll make it absolutely clear. I am talking about the chromosomal determination of male and female. Really what we're talking about is whether they're reproductively male or reproductively female, as in what reproductive systems do they have and do they produce sperm or eggs. That's what we're talking about, the sort of classical interpretation of what gender is from a biological point of view. Now, rather than being determined by a single gene, like all the other examples, your gender is determined by entire chromosomes. Now, remember, your chromosomes come in homologous pairs. You've got one from your mum, one from your dad, or one from each set. So you've got 23 from each parent, but they're effectively the 23 from your mum, 23 from your dad, all match each other and make pairs. Now, when you've got a homologous pair, and you should recognize this diagram from the meiosis video, the two that come from one parent, they're identical to each other. They have identical genes. Same on the other one, but they're almost identical in the separate chromosomes of the homologous pair, because they can carry different alleles. This is what's been at the root of everything we've done in genetics. Now, in your homologous pairs, because you have 46 chromosomes, we can say that they come as 23 pairs, 23 from one parent, 23 from the other parent. But the 23rd pair, if you sort them all out and look at them, the 23rd pair, the last pair, will determine your gender. If you're female, the 2 chromosomes will be 2 X chromosomes. And if you're male, you'll have an X chromosome and what's called a Y chromosome, which is a bit different. It carries some different genes on it. Okay. And if you've got an X and an X as your 23rd pair, as in you've got an X from your mum and an X from your dad, you're going to be a girl. If you've got an X from your mum and a Y from your dad, you're going to be a boy. Now, that makes the genetics fairly straightforward, because the Y kind of acts like a dominant gene. And if you've got one Y chromosome, it will make you a male. Okay, so if we still break it down to our basic same profile that we've done before, mother will be XX, the father will be XY. That means when those chromosomes are separated by meiosis, the mother is going to provide only X chromosomes to the offspring, but the father could provide an X or a Y. It's a 50-50 chance, because in any one cell, he has an X and a Y. So when they're separated, half the gametes will carry X's, half will carry Y. Okay. Now, because of that half and half, it makes it quite straightforward. If the father gives an X chromosome and the mother's had to give an X, you'll get XX children. If the father gives a Y and the mother still has to give an X, you'll get XY children. So it breaks down really simply. Look, you get 2 girls and you get 2 boys in the probability. You get a 2-to-2 ratio or a 50-50 split. Now, that makes life very simple from an evolutionary point of view, because it's the optimal ratio for creating new generations. What it means by probability, there ought to be as many females as males, which means you get as many potential pairings, producing as many individuals in the next generation. So this is just a very clever, simple way that we've evolved a system to make sure we can create as many individuals in each generation as we can going forward. So it's a pretty simple system, but it's just one to add to the ones that you need to know. Now, from this, I want you to go on to the evolution videos. This is now genetics finished, but if you need to go back through any of these crosses, if they haven't made sense, please go back, as guaranteed there will be a question on them, and it can be worth up to about 8 marks in total for all the parts of the genetics question. But go on from here to evolution is how genetics have changed over millions of years to create more and more advantageous individuals with better and better adaptations. The first one of those is evolution 1, which just looks at variation, how varied all of the organisms and life forms on the planet can be. We focus in that on the concept of genetics versus environment to create individualism in new generations. As always, please do subscribe to my channel on YouTube if you haven't done so already. Like this video if you find it useful. Go and have a look at some of my other ones if you haven't been following my course in its entirety as well. I've also got a Facebook channel with loads of extra bits of biology to reinforce and add to your course, and hopefully stimulate a bit more interest in biology as a subject. Thank you.